Hey everyone, it's Mr. Fabeck here, and this is Fabiology Translation, which is the next unit in our DNA segment here, and this is all about protein synthesis. So when we think about proteins, we want to think about meat, beef, fish, chicken, pork, anywhere that we can get those essential amino acids, the amino acids that we cannot gain naturally, so we have to have an outside food source. So even thinking about weightlifting or exercise and fitness, there's all these different protein shakes out there, and if you read them, it always talks about branched chain amino acids. And so what does that mean, and what do we do once we take it in our body? Why do we even need it? And essentially it comes to this. This is the idea. We've been talking about taking our DNA and our nucleus and transcribing it into a language that the ribosomes can read. So we're going to translate that language and make our protein, which will be used in our body for a variety of reasons. So by the end of today, you're going to be able to explain and draw a picture of translation and perform translation. Again, that's protein synthesis from a code of DNA. So you know you mastered it if I can give you any sort of code, whether it's DNA, RNA, tRNA, which we'll learn a little bit about, or an amino acid code, and you can work any language. Okay, that's when you know you really mastered it. So if you can go from start to finish, you're in good shape. So let's just start, because we haven't talked too much. Proteins. Proteins are large molecules formed by amino acids. So if we think about the whole protein in general, they can be very complex. There's 20 different types of amino acids. So if we look at a protein chain or a peptide, you can see that wherever it says R1, that's where the chemical structure can change, and that's what makes this an amino acid. And when you strike them all together, then you make a protein chain. So that you could be like Ronnie Coleman, Mr. Olympia, I think he's a 7-time Olympian, and just build those muscles. Or most of the chemical reactions in our body are carried out by enzymes, which are proteins, and repair tissue. So thinking about grow, repair, and replace. Any sort of muscles, ligaments, skin cells, blood clots, they all deal with proteins. And so we need them, and we're constantly doing protein synthesis. So here's the new stuff. This is translation. So officially, translation is the process of reading the mRNA code to create a protein. So I know that the ribosomes don't really read, right, like you and I would read, but they're reading a code. They're taking in the letters, and they're translating that code into something that the ribosome knows. So think of it like language, okay? Well, think of it like language. So we have a couple different parts. We have rRNA, which is ribosomal RNA, and tRNA, which is transfer RNA, which is going to bring us those amino acids and transfer it into the chain. So here we go. The tRNA holds one amino acid. So when we think about what's called an anticodon, it's at the bottom of tRNA, so it looks like just like that, and it's going to pair with the codon. So the codon is what the ribosome reads in order to synthesize it all together. And this will make more sense once we start to actually do some code here, so don't stress too much right now. But when we have that message from the mRNA, from the transcription, we're going to send it into the cytoplasm, and then that messenger RNA is going to go through the ribosome, and we're going to use tRNA or transfer RNA to deliver that amino acid. And think of it, it's like transferring the baton in a relay race. It's going down the line, and the ribosome's kind of stitching it all together or stringing it together to make a complex protein code. So again, the anticodon's on the bottom, and it's the opposite of whatever the codon would be. So if it's AAG, which is the anticodon, the actual message is going to be UUA. Okay, so we'll get into that again a little bit at the end of the video, but don't stress, but just a little preview. So when we make a protein, we're going to need to have our messenger RNA from transcription, and we're only looking at the codons. Okay, so we're just reading the codons. The amino acids are strung together by that tRNA and the ribosome, okay? And again, there's lots of amino acids that make up a protein, okay? Protein is a polymer. We could have hundreds, we could have 50, we could have just a few, okay? But in general, there's a lot of amino acids that are strung together to make that protein work. So Ribosomal RNA, or rRNA, looks like that. It's it helps go to the ribosome and kind of makes a nice little seal, a little hole that the messenger RNA can go through. And ultimately, it helps us seal everything together. So once the tRNA transfers it, we're going to use our rRNA and our ribosome RNA, or excuse me, our ribosome to help seal everything together. It's kind of like the person who's going to help tape it and glue it together. So this is what it looks like. So if we have a transcribed code, so that's our messenger RNA, and we know it's messenger RNA because it's single-stranded, we can see our tRNAs coming in, and it's trying to match the anticodon with the codon. Remember, A to U, G to C still, and we have our rRNA coming in to help seal it together. So as we go through, we're going letter by letter, and you have to imagine this is going way more fast than we're seeing here. And we're just going through blah, 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 blah. Here we go. And as we go through the entire sequence, we're starting to string that all together until it finally reads a code that says stop. And then there's a little blue monster in your body. No, I'm just kidding. That's just a fun way to say we're done. Okay, we're done with all this junk DNA. And we can move on and let this ribosome, or excuse me, let this protein either go out of the body to a different cell, or to maybe help us repair tissue or build those muscles. So here's how we read it in our class. As a biologist, we want to think about what does that code mean. So there's a couple different approaches. This is what we call a codon chart. And as a ribosome, you're going to read in groups of 3. So when we look at that first code, like on here, we had AUG. Well, if I look at my codon chart, I have A, my second base is U, and my last base is G, so AUG is a start codon, also known as methionine. So every time we look at a protein, it always starts with that methionine amino acid. And then if you also look, we have a few stop codons. So how does the ribosome know when to stop? It stops when it says UUA, UGA, or UAG. These are our stop codons. And what's interesting is you have to think about, now remember there's only 20 amino acids, and so some of them have multiple codes, like phenylalanine here has UUU, or it could be UUC. Serine, you have a variety of combinations here, and so no matter what the code is, we can still get a variety of proteins, and we might even make the same protein with some slight letter changes. So when we think about mutations, sometimes it doesn't make a big deal, because if you just had one letter different here, it might still make the same protein. So we may not see an effect on it. And then here's another way that you could read codon charts. So really, whatever your brain works, that's the one you should work with. It's the same idea. If I want to know what that first one is, I got A, and then I go to U, and G, that's methionine. Or maybe I have UUA. I go UUA, well, that's leucine. Okay, so I'm starting in the middle and I'm branching out until I get to whatever amino acid I have, and it goes just around the wheel like that. So whatever works for you, you just decide. And so if we want to do some protein synthesis of our own, here we go. I have my DNA code. Now remember, the T's, the thymine, does not exist in mRNA code. So after I transcribe it in the nucleus, I have now the code that we can actually use in the ribosome. So you gotta think it's language again. So the DNA speaks, like, let's say, white mung, and the ribosome speaks green mung. Now there's some similarities, however there's a few things different. So once we do that, then we can think about what's going to match up with AUG. So the way I do that is I would have my tRNA, my anticodon there, okay, to show that this amino acid is attached to the anticodon, and then I can get my protein code. So I'd go AUG, I go to my codon chart, I'd see that there. If I want to back up, my next one was, ACU. I go ACU, there's threonine, I would put that down, and I can just then simplify it into 3 letters, but go through my entire code until I see the stop codon, and that's my protein chain. And that's what you want to be able to do. And if you can go from amino acid all the way back to DNA, or if maybe I just give you that mRNA code and you can go all the way to the amino acid code, you are mastering this idea of being protein of doing protein synthesis, and you, my friend, are then going to be a ribosome of your own. So here's a couple ideas, a couple practice problems. Try working backwards. Try starting from the beginning, or make your own DNA code. Just choose a bunch of random letters and then do that translation. So thank you very much, and come back to class with some questions. Have a good one, guys, and start reading those codons.